No. So the next talk is going to be by Tor Wager. So the next talk is going to be by Tor Wager. No. So the next talk is going to be by Tor Wager. No. So the next talk is going to be by Tor Wager. No. So the next talk is going to be by Tor Wager. No. So the next talk is going to be by Tor Wager. No. So the next talk is going to be by Tor Wager. No. Going to be by Tor Wager. So the next talk is no. Going to be by Tor Wager. So the next talk is no. Going to be by Tor Wager. So the next talk is no. Going to be by Tor Wager. So the next talk is no. Going to be by Tor Wager.
Put in other words, where's the pain? And we still don't know this. And so we think about constructing any of our models, we should think about what we'd like to know and we'd like to conclude. And what we'd like to know has to do with two concepts. One is representation and one is biomarkers. And I'll define a representation for present purposes as a causal and obligatory physical basis for a mental experience or another kind of information structure or process. So we heard from Martin about causality. So we know what that means, it's sort of with several definitions. And really what it means for me now is if, for example, this brain process happens, you feel pain. If it doesn't happen, you don't feel pain. If you could intervene and stop that process from happening, the pain is stopped. Like, that's what it works. Of course, we can't measure those things directly, as Rosalind just said as well. What we can do is we can develop biomarkers, which is an objectively measurable process or measure that indicates a mental experience. And so biomarkers, I'll use patterns of brain activity as potential biomarkers. And in fact, every brain imaging study does this in some way or another, for the most part, if you want to say anything about psychology. But some characteristics of biomarkers, they should be at a minimum sensitive and specific for the process that they're intended to measure. And no matter what kind of model you're using, a graph theory model or anything else, then this is a direction that we need to pursue to establish the sensitivity and specificity. So brain biomarkers are gateways to mental representation, because if you don't have a good marker that tracks the thing that your outcome, then you can't understand its representation. It's not a measure of its representation. But of course, it could be a marker, and that's only the starting point for establishing representation. So I'll talk a little bit, take a step back and talk a little bit about why we should be doing all this stuff in the first place, and then a little bit about how to do it in a very general approach that can apply across models, and I'll give you an example from pain research. So why? Well, so let's stop and reflect for a moment about why you do science, especially brain science, or why should we be doing brain science? And for many of us, there are many reasons, but for many of us it goes back to this idea that we like it to improve human health or improve life in some way. And we all do very technical things, but it's actually really nice to keep this in mind. And so what do we need to do to connect our models and our methods to human health and to outcomes that everybody can understand and care about? And so if I could summarize, perhaps a little bit glibly, the first 20 years of neuroimaging this way, there are many findings. And this is an example of 163 studies of emotion, perception in yellow, experience in blue, and you can find results all over the brain. So, and we have many, many findings, increasing in complexity. Now there's over 1,000 neuroimaging studies of affective and clinical processes. We have an accumulation of findings, but I don't believe, apart from a few very limited cases, we have any markers or diagnostic tests that have made it into practice. So this is a part of our job. And one way of summarizing this is the title of this article by Tom Insel, the director of NIMH and colleagues, who said, why has it taken so long for biological psychiatry, it applies to all of us as well, to develop clinical tests, and what do we do about it? So they outline some of the issues. I'm going to go through 4 things that our models can help us do as we move them from models to translational work. One is it can help us understand the role of the brain in health in more precise terms. Many people don't believe you need to understand the brain to understand pain at all, or measure pain, for example. Number 2, it can establish concepts of disorders that are grounded in the brain. Better diagnosis and treatment. I'll talk a little bit about each of these things. 3, it can help us to link animal work to human health and disease, and thereby capitalize on and leverage really a vast neuroscience that's not represented heavily at this conference, but is very important in terms of driving treatments forward. And finally, it can provide objective measures of things like pain and other outcomes too, health risks, performance outcomes. So I'll say a little bit more about what I mean about each of these things. So many of us might believe that behavior is important for health, and in fact in many areas, in cardiovascular disease, in pain management, in the management of almost any disorder you can think of, actually, behavior is really important. But we spend a tremendous amount of money on pharmaceutical sales. So we don't really believe this, because what we're mostly looking for in terms of where the money's going is drug development, $235 billion. And this is the pharma R&D budget from 2004, about $90 billion. And about half of that was spent on actual R&D, and half of it was spent on marketing. And if you compare that to the NIH budget on all kinds of behavioral sciences, decision-making and health, and that's about $5 billion. So we're being outspent about 18 to 1. So most research is really directed toward molecular and genetic causes for a variety of reasons, even when we know that behavior and the role of the mind is very important in all these disorders here: heart disease, lung cancer, pain, depression, anxiety. So I'll go to the second one. New concepts for disorders. Medicine has a history of forming diagnostic categories based on available information. And that available information has been symptoms. So there used to be a category called disorders of the stomach, with all kinds of problems related to your stomach. I mean, that made sense at the time, but of course you can't treat. There's no one treatment for disorders of the stomach. There's no one cause. And so it's not very useful as a category at all. And this is true of other kinds of disorders, phlegmatic disorders, and even things that we have that are considered disorders today. So you could have chronic pain, and that's sort of one category of disorder, but this might be lots of different things all mixed together. Depression is another example. So it turns out that two people can be depressed, and they can have non-overlapping sets of symptoms. So they can have completely different symptoms, but they're both depressed. And then we're looking for brain correlates of depression as a whole. Well, there aren't any. The hope is what the brain can do for us is help us to establish more refined categories, diagnostic categories, that hew more closely to the neurophysiology itself. So that's part of our job. 3, there's a lot to say about this, and this is really a nice thing to keep in mind. So this is a typical treatment development cycle in pharma. So we've got some disease feature, like pain. Okay. And then we have an animal model for that, where they seem to show the same behavior. And then we can look at the molecular mechanisms, you can understand the system, you can try develop different treatments, which leads to testing in animals, testing in humans, and hopefully then something that actually gets marketed to humans to work for treating pain. Unfortunately, this has not worked very well, and this failure has been one of the big stories of our time, really. The problem is, there's a couple of problems. One is that disease features like pain are not biological categories. And so looking for mechanisms, establishing this correspondence here, is arguably maybe not a great thing to do. And of course, mice, where you can do a lot of stuff, are not humans. And pain behavior in a mouse is not human pain. So there are many different kinds of human pain, and all the animal measures are really measuring something that's probably very different than what the average patient actually goes through. So then this entire cycle is flawed from the start. And what happens then, if you don't have the right homology there between the human and animal features and behavior, then what happens is the trial fails there. And this is probably maybe $100 million later, let's say. So huge problems, and this has caused a lot of debate and controversy, and it's also caused companies to simply just pull out of brain research and pull out of trying to develop treatments in some areas where it's just not cost effective. So we all like to blame the pharma companies, but really it's all of our responsibility to do something interesting to help. So neuroimaging-based biomarkers, what can they do for us? If you look at brain patterns or models or features, we can link those, on the one hand, to human outcomes, to pain, for example, cognitive performance, and on the other side, we can link those to models of neural circuits and neural measures in animals. So the brain, our brain measures themselves provide the bridge, allowing us to develop treatments. So this is what a revised treatment development cycle might look like, and this is, you can call this reverse translation, where we take human pain, we map it to human brain patterns, and we map that to what are the animal systems involved, and then what's a good animal model that targets that system, and then we can develop a treatment. It's a lot of work, but I think we need to do that to move forward. So how can biomarkers help us? They can measure components of mental phenomena like pain that we really haven't had measures for before, and many people believe they're still not measures. And these measures are not pain. Pain is a subjective experience, like many kinds of symptoms and complaints. And brain measures are not pain, but what they are is they're measures of neurophysiological processes that can give rise to pain and other kinds of disease processes, depression, etc. They're less subject to decision biases and to differences across cultures. They're closer to the biological systems that generate that disease process. They're more likely to map onto specific mechanisms and treatments. Can't we just ask people how they feel? Yes, we can ask people how they feel, but I think I've tried to make the case that it's really not good enough. And this example, I won't go into details about it, but really basically, if you give people a different rating scale, then their pain, can double or be cut in half, right? So how much you rate pain on a 1 to 10 scale is not an absolute measure. People use the scale differently. And this has resulted in a lot of problems that really plague our societies now. And one of those is that people in pain are not often believed. So there's lots of popular articles about this here, but in a nutshell, if you are a minority in the US at least, and other places as well probably, if you're a woman, you have to report more pain to get the same amount of treatment. If you're a minority, you're likely that people won't believe your pain, and you'll get referred to drug testing and then to counseling, instead of treated in the way that somebody who has got more money and is the right socioeconomic class and race would have been. So these things really play out, and then part of it is because we don't have these measures. So brain science, I really believe, can help. But like I said, we have lots of findings, and what we need is models that can be used for translational purposes. One of the biggest obstacles to believing that we have biomarkers for mental events like reward and value and pain is the belief that we already have them. So amygdala activity is often taken as a response to aversive images. Pain processing in response to noxious heat is taken as a measure of pain, but there really aren't biomarkers for any of these processes yet. And no matter what kind of model or technique you use, you can apply these principles that I'm telling you now. So these results are not biomarkers. Why not? Well, for one thing, we have a problem of definition and replication. We don't agree on precisely what the patterns are, which voxels, or applied to a DCM, what exactly the specification of the model is from one study to the next. So there's a lack of exact replication. Everyone's doing different things, and nobody's using other people's stuff, right? Other people's results, really. Number two, we have a problem of application to individual cases. We often do group studies with group results, and that's a proof of concept that there's some relationship between, say, brain and depression, but what we really need to do is have models that apply to individuals. And three, we have a problem of diagnostic value. And the problem is essentially that it's not often assessed. So if I think of this in Bayesian terms, what's the probability of experiencing a psychological event like pain, given a brain marker, some pattern? We have a problem of sensitivity. So I might know that there's some association statistically, but I don't know if that brain measure is really large enough to capture the phenomenon, to explain the task or the outcome that I'm studying. And that, in Bayesian terms, again, you can think of this as the probability of observing this brain marker given the fact that you're in pain. And how strong is that really? Secondly, we have a problem of specificity, which is we don't know if the observed patterns, models, findings are specific enough to be useful as biomarkers. So we don't know what's the probability of observing this thing that I'm measuring in the brain in the absence of pain or the psychological event. So establishing this is a tall order, but it is possible. And it's possible by sharing results and validating models across studies. So this is the approach that we can use for all of these techniques. First, we can optimize a pattern, a marker or a brain pattern or model, and we can try to maximize its sensitivity, its specificity, its interpretability, how much it matches results from animal models, for example. When we have a provisional pattern, we can then replicate that pattern. And really, this is a process of assessing its generalizability across new individuals. If I take pattern X that predicts pain, and I plug it in and test a new individual that I haven't tested before with this, how well does it predictive of pain? So we can assess generalizability across individuals, across studies, across populations. And many over the last 40 years, there have been many attempts to develop biomarkers in various ways, from blood biomarkers for alcohol, for example, that have failed after initial excitement because they didn't turn out to translate to new populations. And so passing these things around, our models and markers and results, is really important. And then we can characterize the properties of this marker across test conditions. So when does it fail? When does it track pain? When does it fail to track pain, for example? Once we do that, then we have a growing, what I'll call a proxy for a representation, right? A representation of pain, for example. And then we can test new treatments. We can test uncertain cases where you're not really sure what somebody actually feels. We can use that to identify subtypes of patients. Everybody's got pain, but there's different neurophysiological basis for that pain. So maybe different treatments, different causes. And we can use that to understand representation and mechanism. So here, in the examples I'll show you next, we'll always develop models under conditions where pain readings are really accurate and reliable, and test them in cases where the pain reports are unreliable or unavailable. So we're always sort of, initially we're using what people tell us as the ground truth. But we don't need to stop there. We're not measuring that. We're measuring then a neurophysiological process, and the more we trust that process, the more we can move beyond self-reports and say something new. Okay, so here's some desirable characteristics for developing a biomarker. It applies to any result that you want to carry through to translation. There should be interpretable metrics for evaluating it. Sensitivity and specificity are a good start. We should be able to apply them to individual patients. There should be interpretable brain features that make sense. I think when I talk to people who work for the National Institutes of Health, for example, many people say, well, all that MVPA pattern machine learning stuff, that didn't really pan out. I say, what are you talking about? And they say, well, all these patterns are like junk all over the brain. I don't know, right? So they really should be patterns that make sense. You can look at the first competitions, the PBIIC competition in 2006, and then subsequent, machine learning competitions produced results that seem to be very predictive, but they don't make sense always, right? And if the winning models don't make sense, people will never be able to trust them and never know when they fail. And then another thing is a marker should be scalable. It should be accurate and it should be usable across scanners and patient groups. We should be able to pass it back and forth relatively easily and test it. And it should respond to known treatments. And generalizability I talked about. And then the last thing is, if you actually are going to use something for a real clinical test, what happens if your scanner is not working this well on this one day? What happens if your EEG electrodes are not quite right? You need positive controls. So you need things built in to the paradigm that says, if this fails, if I don't see this brain marker or feature, then my test isn't valid. And this is standard in biological assays. So molecular biology is great at this, I think, and they would be nowhere without these kinds of tests. So we need to start thinking a little bit like molecular biologists in that way. So here is an example. So you say, why do I need to study pain? We're going to develop a marker, a pattern that predicts pain, acute pain. And why would you need to? You can already tell if people feel pain, right? Well, I'm going to introduce you to Philip Pizzo, who is former dean of Stanford. Got a great resume for pain, treated pediatric pain for 30 years in kids. And he is also the chair of the Institute of Medicine Committee on Relieving Pain in America. So if anybody knows about pain, it's him. He just wrote an article recently in the New England Journal of Medicine, an op-ed piece. He said, I thought I understood pain until I was at the airport putting a bag onto a conveyor belt, and all of a sudden something in my butt happened. And he ends up with pain. And of course he thinks it's going to go away, like all of us do, and then it gets worse, and then it doesn't go away, and it becomes neuropathic pain. And then his doctors, and this is the guy who's, a lot of respect, right? This is the doctor, doctor himself, and people start going, are you kind of depressed? Maybe there's something, maybe you're not happy at work. How's the dean job going? I don't know, right? And so they don't know what to do, and all of a sudden, they stop believing his pain is real. Only because he's got so much, I think, money and resources behind him, perhaps. They did a number of tests, some atypical tests. They found an unusual branch of his nerve that was something that they could go in and surgically alter, and then his pain got better. But if they hadn't done all those tests, most of us out here, if this were us, we would be on psychiatric drugs, and we wouldn't be helped, right? So there's a lot of value in just saying, here's something in your brain that really tracks, yes, you have ongoing pain right now, and we're pretty sure it's coming from your body and not some other system. That's one starting place. So here's a biomarker development process for that we've been engaged in for predicting pain in acute experimental settings. At first, we're using that to translate that into clinical context. So the setup is pretty simple. We got 20 people, thermal pain in the left arm, 12 trials at each of 4 temperatures. They're calibrated for that person. And we can do lots of fancy things, but really looking at a standard general linear model response to heat on a person by temperature basis, we look at pain-related activity, works pretty well. And I'm sure we can improve it, but this is a good place to start because it's quite scalable, essentially. And we'll take a multivariate approach. Other people after me will talk more about machine learning and pattern recognition, so I'm just going to be very basic here. We manipulate the noxious input by increasing the intensity. We're measuring brain activity in many regions at once, now about 100,000 voxels every second or so. And then those are combined into a predictive model. So here's a predictive map, combines information in a regression model from all these regions together to say what pattern can we identify that predicts pain reports. We'll use a linear pattern so it's interpretable, and a couple of tricks from machine learning that I think are important. One of them is the idea that we have many voxels, many predictors, so we're going to use machine learning to stabilize the maps. I won't talk about the details now of how we do that, but there are many ways to do it. And then the second idea is testing generalization. So we're always going to train on some individuals and test on other individuals. So everything that we're identifying in this map is really something that's conserved across people. And then across studies, we can ask how does it work in different populations and different samples. So this is what the map looks like, and I won't go into too many details, but I'll say 2 things. One is we see pain-related responses that are predictive of pain reports trial by trial in really all the areas you'd expect from animal physiology and from human physiology of pain. People have done a lot of operative surgical things to try to affect pain and measure it. So we have a fair amount of background knowledge. This is a good map. It's interpretable. Secondly, we know now that the pattern itself is actually quite important. It's not just that you have activity in the anterior cingulate there, but it's the actual profile that's really important, and it helps establish its specificity. So we can apply that to individual person data and generate a prediction about pain intensity, and we can test that in various ways. So this is what it looks like in this first study. We've got 4 different stimulus intensity levels, and what we can see is only during the actual heat, only when it's painful does it go up with stimulus intensity. There's no response to anticipation of pain, threat cues, pain recall, and we can turn those continuous predictions about pain for a given person into a test. So we can do a forced choice test where we take a painful and a warm image, and we compare those things, and we see which is more painful, or a pain versus a threat cue, or a pain versus recalling and judging pain, and we're quite accurate. We're above 90% in all the cases here in cross-validated analyses. Of course, it's easy to fool yourself with cross-validation, and we need to know how it generalizes to new samples. So this is an example from a prospective application of that pattern to, again, heat pain from a different study, a different sample, a different scanner. And here there are 6 temperatures, so you can see that this is the response here, which is really the dot product of our pattern template and each individual brain image that we're feeding in. So we can apply that to people, we can apply it to trials, we can apply it to time series images. So here it's just the average of a few trials per condition, 8 to 11 trials per condition. And what you see here is that this signature response then becomes one outcome measure that we can test, and it goes up nonlinearly as a function of the temperature, just like pain does. And I won't belabor the details here, but we can look at this as a function of whether people said it was painful and non-painful in each trial, and ask, is it really tracking pain and not just the temperature or attention or stimulus discriminability? And in a nutshell, it really starts to respond and shows a response with increasing levels of pain, but not touch, even though touch is discriminable. So that's the idea. And we can feed it in, and we can be pretty accurate, again, above 90% accurate at discriminating which is the more painful thing for a given person, as long as the stimuli are moderately different in pain. 1 degree Celsius is enough to get to 90% accuracy. So it works pretty well, better than I ever thought. So now we can ask, what's the NPS really measuring? We could be measuring specific subtypes of pain, or any kind of somatic pain, or maybe anything that's negative, or maybe anything arousing, or anything salient. And this is where we have to know, what are we measuring a representation of? We have a proxy for a representation of something, but what is it of? What do we have to do? We have to find things that are a lot like pain, but that are not pain, right? But that are really similar. Fortunately for you, my heartbreaking movie of social rejection didn't work, so I'm going to move on. But rejection is one thing that we can compare to physical pain because it's aversive, it's arousing, it hurts. So to speak. So here in this second study, we have 60 people, and they were all romantically rejected. So they looked at pictures of their ex-partners that they brought in and pictures of a close friend. And they also experienced painful and non-painful heat. And so then the setup is simple. What we're going to do is we're going to ask, does the marker respond to pain, and does it respond to the rejectors? And if so, it's not tracking physical pain specifically. But if it's specific to physical pain, then we can establish its specificity. It's not just something negative. And what makes this an interesting test case is because when we look at the areas that are activated in common for physical pain and rejection, they're very similar. They activate all the same areas, broadly speaking, that we care about, including anterior cingulate and insula, and even the posterior insula and secondary somatosensory cortex. And that's important because there aren't very many other things that activate somatosensory cortex, but these rejector photos do. So then we can apply our test, and this is again the same marker that we developed in the earlier studies, and we can test it on the hot versus warm, and here it's 90% accurate at picking out which is the more painful thing. That's the average of 8 trials per condition, per person. And we can also then apply it to rejector, the rejector case. And Brad Pitt's a rejector, that's why I put him there. And here it's not different from chance. So okay, so it's not just anything negative, it's not this thing that looks a lot like physical pain, it's something that's more specific. And here's the expression of that marker. Here is when it's actually painful, here's warm, and then there's the rejector and the friend, and there's the threshold that we established in the first study as part of our model. Okay. So let's just summarize this. And we've done this and more. And what this is a multi-study process of taking one result or pattern and sharing it. And now we've shared it with many other labs, and we're interested in sharing it with more labs to test different conditions. What's not pain but looks like pain? Maybe it's, air hunger, or something else. And what is it? What kinds of pain does it really capture and track? Does it track visceral pain, for example? Does it track clinical pain? And there's an ongoing process. We can't do it all in one study. And just to summarize this then, we have evidence for specificity, that there's no response to threat cues and pain recall and rejector images, and looking at other people in pain, looking at emotional images which are disgusting or arousing, so lots of other things. We do see transfer across types of acute experimental pain, like shock and mechanical pain, that's my timer, and visceral pain, it turns out. We see transfer across body sites, arm, foot, thumb, leg. We see some treatment effects when we know there are drugs that should affect pain. And we have a lot more to go, but we do this piece by piece because the process of sort of annotating this, is a long process. And the more promising it is, then the more we can invest resources and essentially try to test it more completely and falsifiable, and falsify it or validate it. So that's a little bit about the process. I hope that we do this with many of our models, no matter what kind of algorithm or graph or whatever you're using. I think this is an approach. And if we think about these characteristics, and I'm just about done, Nico, thank you, what we've done is provided some evidence, really, I think, for all of these things to some degree. And we can always do better. But that's a way to start thinking about it, what's good and what we want. All right. And finally, I think, for further reading, there's a biomarker definitions working group, and David Borsick has written a lot of stuff about biomarkers, and this is the idea. You start with a preliminary biomarker, and I think we've gone through some initial stages of biomarker development and validation. But of course, to know that it's going to work for everybody out there, we need a lot more tests, So then as the most promising models and markers are developed, we can keep testing them, and then they can be scaled up to larger and larger tests and use cases. So that's the idea. So to summarize, I think human imaging plays a key role in translational science. This is something we all could do to keep in mind what this end goal is. To make it useful, we have to identify markers with defined diagnostic properties. We have to pass them around and share them and test them across different contexts and different populations. And that's a way forward. So with that, I'm going to stop there. And thank you very much. And this is my lab, and thanks to my lab and all their help as well. Thank you.